Thanks, Flora. So today I'd really like to share with you both the general approach we take to using data to address questions, lots of public data as well as data from collaborators, and then the specifics about one of these projects where the data is coming from a long-standing collaboration with the lab of Ron Mackay. Um, so I'll get into that after I give you the general outline of our approach. So this is where we start, which is I run an entirely computational lab. So I'm either collaborating closely with individual PIs and groups to get data and run projects, well, usually and collecting data from public sources that are focused on particular cell or disease contexts. So I list a couple of these contexts that I've studied in most detail at the bottom, but for any one of these, and indeed most fields in biomedicine today, there's a huge amount of public data available to begin to understand whatever it is you might be studying in that particular context, even if you're not using multiomics data. So, across species, model systems here, I just list mammals, but of course there's Drosophila, worms, fish, everything we study, depending on the cell or organ you're studying. And increasingly today, there's not only these in vivo models and in vivo human tissue, but organoids in vitro and even 2D or other lab on a chip kind of scenarios. So a huge amount of different perspectives from cells and tissues and animals grown and experimented in different ways. In addition, there are different levels of analysis. You can have bulk data that comes from a piece of tissue. You can sort cells and then sequence them. Laser microdissection is super interesting. The next level past that are some of the amazing spatial transcriptomic and spatial epigenetic modalities that are out there today. And there's a ton of single cell RNA sequencing, and much of what we look at today will be bulk and single cell RNA sequencing. And as I said, there's lots of these different modalities as well. So you're look at RNA, you can ATAC to see chromatin accessibility, ChIP-seq to look at specific epigenetic marks, DNA methylation, chromosome conformation data. So there's a lot of data out there, but most of it is not available to biologists who do not code, that don't have bioinformatics expertise. So a lot of what we do is directed at making it available to people. So that's step one, is getting the data together. So the second piece of this general approach that we take is decomposition and projection. So what do those two terms mean in this context? In the simplest form, decomposition means taking one experiment, whether it's an RNA-seq experiment or otherwise, and breaking it into pieces that are biologically meaningful. So that can be as simple as a packet of genes that runs together. Their expression is correlated in the experiment you're looking at. There's traditional linear methods, PCA, NMFs, non-negative matrix factorization, that we use a lot. And for years now, I've been collaborating with Brian Caffo here at the Biostats department at Hopkins, and with my first graduate student, Genevieve Stein O'Brien, who's now a PI here at Hopkins as well. And we've been really working on developing methods, not only to decompose one data set and then look at the pieces you get from that data set in another one, which we usually call projection. So looking at that packet of genes you might find traveling together in your first data set, well, what are they doing in this other related data set, whether it's an in vitro model of the first one or something like that. But the other step is to say, well, we also want to decompose things when we're actually looking at 5 or 10 matrices at a time. So that's the joint decomposition methods that are in some of these references here. Okay. The third piece then, once you get all the data together and you have these tools to decompose data sets and look at those pieces across many data sets, is to do this in an environment where not only the person with the coding skills can look at this and explore this space, but where any biologist can do it. So I've been working for years now with Seth Ament, Anup Maharkar, Rona Herzano across town at the University of Maryland in the Institute for Genome Sciences, um, and Sean Adkins and Joshua Orvis are some of the web developers. So a lot of different people with a lot of different skills coming together to bring data resources and access to some of these projection tools online. And actually, the pictures at the right here are screen captures from that system, where each one of those boxes is a single data set or a single experiment, whether it's bulk RNA sequencing or these are mostly single cell. So we're now trying to say, okay, for at least some of these particular focus areas that we're interested in, we have collected a large amount of data. And we're in the neocortical development environment, for instance, we have nearly 200 studies, somewhere between 30 and 40 million individual samples, where a sample can be an individual cell and single cell or a bulk sample. So we really have a good chunk of the data out there that is relevant to these systems for a very small number of biologically driven realms or regions of study. But once you get that, firstly, biologists can surf those data sets one gene at a time. That's what the screen captures are from, looking at the eomesendermin gene in neurogenesis. But then they can also look at these decomposed pieces. We don't have the decomposition online yet, but we'd love to get that going. But what we do have is if you see a principal component or a set of 20 genes that someone publishes or that you have found in your own work, looking at the activation of something or the turning off or turning on of a gene, and you want to see what's happening to that particular packet of genes across the whole data collection, that's just a few clicks away in the environment. Now, it's only a few clicks away because the work has been done to collect the data and put it there, and then enable the sort of mathematics of projection to reach out to each one of those data sets using your particular gene set of interest. But it's really, again, the idea is putting this in the hands of cell biologists of many different kinds, not just the minority of biologists that can code. So, please reach out to me or these websites here. These links will be live in the slides. I can send them to you to really surf these data sets. And yeah, that's where we're going. So that's the general approach. You got to get the data together, the tools to break it apart, and then really the tools to be able to look at it. And I obviously myself can code, but I use our environment every day to look at data sets. If I'm going to look at a particular gene or a particular complex gene signature across 50 data sets, I'm not going to code up a visualization for 50 individual data sets and different colors for it and da 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 da. It simply takes too much time. So once we have it automated, you can begin to ask very focused questions in silico once you have a data collection and gene signatures of interest. Okay, so now jumping into the particular example here of connecting the dynamic emergence of lineages from pluripotent stem cells in vitro to early mammalian embryonic development. So again, as I mentioned, and as it says along the bottom here, this is results from a long collaboration with Ron Mackay and many in his group. Sil-Kee Kim and Seung-Mae Seo are also here, and they're the first authors on this paper. So a story that I heard Seung-Mae Seo tell recently, actually on Martin Pear's podcast, was that when she joined Ron's lab, Sil-Kee Kim was already there. And the first thing she remembers seeing is Sil-Kee explaining to her how this one cell line I4 and this other cell line SAO1 behaved very differently and repeatedly so. Anytime you grew them, and no matter how what passage it was or the plating density, that IO4 was more efficient at producing the mesendodermal lineage, while SAO1 was more efficient at producing the neural lineage. And that's actually what we're looking at here. We're looking at 4 days after the cells, the pluripotent stem cells, were treated with NSB, which is one version of the dual SMAD inhibition protocol to induce the neural lineage. from pluripotent stem cells. And as you can see, not only is IO4 and SAO1 quite different, they're sort of at the two ends of the spectrum, but the other cell lines are in between them, each with a different level of efficiency or speed of turning on the neural lineage from pluripotency. So this is some cell imaging, but we also did bulk RNA sequencing. So for each of the 6 cell lines, we looked at pluripotency, BMP treatment, which is the induction of the mesendodermal lineage, and then the NSB treatment or the dual SMAD that induces the neuroectodermal lineage. So black here is pluripotency. So this is a PCA plot of that bulk RNA sequencing experiment. So in pluripotency, you kind of stay in the same realm. And something we have that most people didn't or don't look at is actually 3 time points in the pluripotency cycle. So if you plate out pluripotent stem cells, let them grow before they hit confluence, you replate them. This is kind of 3 time points in that cycle where these small dots are day 2 and then 4 and 6 will get bigger. So you can think of this as like a little swirling pool of the pluripotency cycle. If you hit them with BMP, they move very quickly. So the first day 2 is right here. If you hit them with the dual SMAD, they move a little slower. So they kind of emerge from that little pool of pluripotency and start to stretch down this way. And just as we saw in the cells themselves in the RNA sequencing data, you can see SAO1 leads this emergence of the neural lineage, while on the other side, when you hit the cells with BMP, it's IO4 who's leading the way. And in fact, that's why there's only 5 small dots here, 5 day 2s. The day 2 for IO4 is actually all the way up here. So it's moving forward much more quickly than the others and ends up ahead of them here as well. So both in many cellular assays, here looking at SOX21, which is the marker for the emergence of the neural lineage, and then the RNA sequencing itself, we saw this spectrum of variation across the cell lines. And the key was that it was stable. Every time Soki or May or anyone grew the I4 line or the SAO1 line, they saw this difference. And I'll mention later also something that's very interesting. One of the lines here, this ES cell line, H9, is very commonly used across the planet. So we started not only to see particular behaviors and particular signatures for these lines, but then we could actually look at data from many different labs to see if these cells were doing the same thing every time they're observed. And indeed, we can see very consistent both cellular and molecular traits that are very stable in these lines. And that's really what we're working toward understanding here. Okay. So these are now 2 patterns that we pulled out of that same experiment. Again, so this is part of that decomposition notion. So this is the overall look at the RNA sequencing experiment, just looking at PC1 and 2. And you could even think of this as a decomposition already, where PC1 is one piece and PC2 is another, where PC1 really separates the mesendodermal lineage, and PC2 is pretty much time moving forward in either neuroectoderm or the mesendodermal lineage here. So we used an NMF to break apart this data set into some more granular pieces. And these are 2 of those pieces. We got a set of 22 patterns from a non-negative matrix factorization called GW COGAPS. And so we're visualizing the data a little bit different here. So to orient you, this is 2, 4, and 6 days. And we have SR is self-renewal, that's the pluripotency. NSB in red is the neural lineage, and BMP in blue is the mesendodermal lineage. So we have the 3 different conditions, the 3 different time points, and the 6 different lines in each one of these panels. So in the 2 panels are 2 different pieces that are where we decompose that we pulled out of the larger RNA-seq experiment. So the first one, this pattern 7, is clearly pluripotency, because you see here all the black lines are very high. Actually, over time in the pluripotency cycle, they descend, but as you replate then, they're back high, and so this would be your pluripotency cycle. If you hit them with BMP, this disappears very quickly and stays off through that whole experiment and would through the life of a committed cell. And in NSB, it's a little bit different. It moves down, but it moves down more slowly, and in particular lines more quickly than others. And this is, again, SAO1 leading the way in that neuroectodermal lineage. Not only does it lead the way in the turning on of that lineage, but in the turning off of its pluripotency signature when traveling in that particular direction. You don't really see the IO4 difference here because everybody by day 2 has turned off pluripotency when they get hit with BMP, the different lines. The second pattern is one that gets turned on during BMP stimulation. It's totally off in pluripotency and in the NSB condition, the neuroectoderm. You can see everybody's turning it on, all 6 lines. 2 of them turn it on with different efficiency. And so it's, again, of interest to us. But the idea here is that we've now taken this experiment across 6 lines and some time to look at the emergence of these 2 lineages. And so that was our decomposition, if you remember the original approach at the beginning. So then what we can do is the projection. So now what we're looking at is an entirely different data set. This is from Piwansala et al. This is a Berti-Göttgen study. It is mouse. It covers day E 6.5 to 8.5, so 2 days of embryonic development in the 20 to 21 days of gestation that it takes a mouse to give birth. This is 6.5 to 8.5 is really when pluripotency is just starting to, or lineages are starting to emerge from pluripotency, gastrulation is starting to happen. And that's what we can see here is the projection of the pluripotency pattern in the top row and the mesendodermal pattern in the bottom row. Where do these gene signatures end up in vivo? And so you can think of each of these signatures as weights for all the genes in the genome. And for the pluripotency signature, for instance, SOX2, OCT4, NANOG, and these kind of genes are going to be really high. The vast majority of genes in the genome are going to be almost 0 weighted because they're not on in pluripotency. But hundreds, maybe even not a couple thousand, will play a part in this signature. And similarly for the mesendodermal pattern, it'll be a different set of genes, but hundreds of these genes will be involved in the emergence of the mesendodermal lineage along this particular temporal trajectory in vitro. So when we take all those weights, we can project them into the in vivo data. So when we project them, it's super important to understand the map. So this is the published UMAP. If you look up this paper, you'll find the same map with different things visualized on it. What are we visualizing here? This is pattern 17, the pluripotency pattern. So if we look at the expression, the aggregate expression of the genes with high weights in this pattern, we see it really lights up these cells back here in this single cell experiment. What is back here? In the guide, we see that this is the pluripotent epiblast. That is before gastrulation, before the emergence of committed lineages in the embryo. And this is now the beginning, the primitive streak here moving outward. But really where the pluripotency signature is high is here in that pluripotent epiblast. And then similarly here, where is the mesendodermal signature highest? It is not on back here, but in fact, cells have to get all the way out here and here before this turns on. So this is actually mesoderm of different flavors up here, and this is endoderm of different flavors down here. So that's, this is not a mesodermal or endodermal specific. It actually spans both. And here we have some of the specific lineages here. So this is cardiac. I think this is mesenchyme up here, and the allantois is over this way. And some of the different endodermal lineages are down here. We didn't label them here, but this is showing you, again, the mapping of the signatures we're getting from the in vitro dynamics of differentiating pluripotent stem cells mapped onto the mouse embryo. Okay. So these are 2 more patterns. And now, instead of looking at pluripotency and mesodermal lineage, we're going to zoom in on the neural lineage that was really the focus of most of the rest of our paper here. So this first pattern, as you can see, is already on at 2 days at differing levels in the different cell lines, SAO1 leading the way again. And in fact, you can see that SAO1 actually does something that none of the other lines do by day 6. It's actually turning off this pattern, this sort of neuroectodermal initiating pattern that includes SOX21 that will that we really that has been and we define further here as a pioneer of the neural lineage in mammals. And as it does go through this process quicker, it actually turns this off, and all of them do, but just later. It actually turns on this late neuroectodermal pattern quicker. So as 15 goes off, this pattern 12 comes on in SAO1. So this is a later pattern in the emergence of the neural lineage that at day 2 is not present. At 4, it starts to whisper, and then 1 or 2 cell lines, and maybe a 3rd or 4th just beginning here, it really takes off at day 6. So just like we projected this into the early mouse embryo for the pluripotency and mesendodermal lineages, we did the same thing here. And again, this is just like the figures from the paper, but now instead of colored by the gene or the lineage they were looking at, it is now colored by the projection of our gene signatures that we've associated with particular lineages and particular temporal dynamics. So this is step 1 and step 2 of the neural lineage. And if you follow the link, the live links that'll come in these slides, you can hit the website and actually request any gene you want, and it'll be displayed in our data as well as the P-Huansala data. And you can also look at these patterns. We have all the patterns loaded into the website, so you can actually do this projection live and look at these. And so what do we see here? Similar to what we saw before for pluripotency in the mesendodermal lineage, we now see that this is indeed the neural lineage. So this is the portion of the UMAP that it's lighting up in the single cell data, and that corresponds to maybe something about here. And so that is what they're calling the rostral neuroectoderm and the midbrain, forebrain, hindbrain. So they didn't try to differentiate the brain regions yet here, but in fact, if you subset that, talking about in a minute, that you can see that there. So this is now showing us that the in vitro signature that's projected into the in vivo data corresponds to what the original authors already identified as the neural lineage. And here, similarly with the second one, it's a little pushed forward. So this is, again, we're moving from pluripotency. We knew from our own data that P15 and P12 were sequential temporally, and we can see the same thing here in vivo, where P12 is lighting up a little more progressed down this neural lineage. Appreciate it. So that's really how we try to, marry the traditional way of presenting data in a figure in a paper that is, of course, not read in hard copy anymore, but we distribute them digitally, but to make that live, where there's a picture showing a single gene or a single process in a data set. And now with a single link, we can get to that same data set, all the underlying complexity of it, and really dig in and look at it. But again, like I said, that really requires not only a lot of back end getting the data ready in places where it's accessible to people, but on the end of the user to beginning to know this map, so that when we see this area light up or this area light up, for instance, all of us that have been doing this work together know this is blood. The earliest progenitors are here, and these are some of the earliest erythropoiesis going on there, so that we can understand these pictures as we begin to look at many, many, many different pieces of them to understand what's going on in any one context. Okay.
Can you make biological sense of some of this? For example, from the pattern to the left to the pattern to the middle? I mean,
yeah, absolutely. So what we've done, so now what we know is that this pattern that we can capture even before 2 days, right? It's on before 2 days in vitro, that basically the first thing pluripotent stem cells start to do when they move toward the neuroectodermal lineage, SOX21 turns on. That's one of the genes we looked at in the cell staining. So we could go through the entire list of genes that's involved in this and know that they're not only what happens to a cell when it's beginning to form the neural lineage in vitro, but we can see very specifically when and where these things are happening in vivo. And again, when we have those specific gene weights, we can see if it's the entire pattern, if it's all of pattern 15, or maybe there's some of pattern 15 that is involved in this signal and some of it is not, because it is an aggregate of all those hundreds of weights we were talking about earlier. This projection is a aggregate of all of them.
But you said that this is not neural, right? What's that cluster that lights up in the mouse brain?
Yeah, this is the beginning of the neural lineage. So it's rostral neuroectoderm, caudal neuroectoderm. What they sort of in a hands-off way said forebrain, midbrain, hindbrain. If you actually look deeper in there, in some of our patterns, you can see those different regions already beginning to form there. But yeah, the green pieces here, and part of that map is right what's lighting up right here and down here.
So what's the difference between the middle and the far right then?
The far right is simply a legend for you to understand what we're seeing here. So this is the power of pattern P15 from in vitro, how powerful it is in each cell in vivo. That's what the middle is. And this is just to guide you to, well, what cells are turning on there. Without knowing the map over here on the right, we wouldn't know what pattern 15 is lighting up. We know from in the in vitro experiment, it's the neuroectoderm, right? It's getting turned on in the NSB. Here, that corresponds in vivo to the beginnings of the neural lineage. And we know that because of the marker genes that they already knew where they were expressed here. And 12 and 15 are very tightly related, not only in vitro, but in vivo. They're sort of overlapping here, and the other one would be right on top of it. But you can see both in vivo and in vitro that is sequential. And again, we could go drill down into which genes are in which of these patterns. These are sort of the canonical telencephalic genes start to come on in this pattern 12, where in pattern 15, it's more pan-neural. If you want to make a neural cell, you need this packet no matter where in the brain or CNS you're wanting to make cells.
Carlo?
Yes.
I have a question. Hi, how are you? I have a question about, because I went on the metadata, like the type of pluripotent stem cells that you are using, and I noticed that basically the first three actually are embryonic stem cells, while the other three are pluri-induced from reprogramming skin fibroblasts, right? The top one. So I wonder if you... Yeah, three of each. Did you try to... It seems like that the embryonic stem cells activate the neuroectoderm better than the reprogrammed one. And did you do any analysis regarding this? Thanks.
Sure. So with the 3 and 3, we really did not try to make a distinction between systematically what an embryonic stem cell was doing versus the induced pluripotent stem cells, because the low N, and we in general did not think that was what was driving these effects. And as we'll see in a second set of 6 lines, we use all induced pluripotent stem cells, and we can see the same sort of range of variation. And by the end of this paper, we use some public data that has bulk sequencing of 300 lines, all IPS lines, and the range of variation or the dimensions along which the variation lies is the same. Whether if you had, 20 ES cell lines and 20 IPS cell lines or more, that there would be a systematic lineage bias in one or another of those types of human pluripotent stem cells, I don't know. I think that's not the central question for us in general. Our central question was across human lines, whether they're ES or IPS, what is that variation? That's not to say there aren't some things buried in there that, get close to sort of naive versus primed pluripotency, and are there other biases there? And does an ES cell sit at a slightly different lineage bias state than an IPS cell? But we didn't really have the numbers to go into it. That's not what we were interested in here. And by and large, the biggest differences and most interesting differences we see, we do not think are necessarily ES versus IPS, but we did not test that particularly systematically.
If I can jump into that,
sure.
did you have at the end enough number to compare variation, intrinsic variation in ES lines versus IPS line? Because if I might remind you, IPS line are originally somatic cells. And somatic cells have somatic mutation and somatic variation that may be different than the one in embryonic stem cells. So I wonder whether, although there may be variation in both, I'd be fascinated to know whether this variation, the extent of this variation is the same in those
Yeah,
two.
I don't think anybody has the numbers, to tell you the truth, simply because of the limited number of human ES
Yes,
lines. Maybe in the mouse that people could, but there's so little motivation right now to create large numbers of ES lines.
Of course. Yeah.
That I think that's going to be a hard question to answer. I don't know, Ron and Soki, if you guys have some ideas on this, please do jump in. But yeah, our focus was not on that here. Okay. So I've showed you a couple.
I'm obliged to say something, Carla,
Yes,
just to say
in,
I'm still
please.
here and listening and enjoying this discussion very much. Yeah, we could go on that topic for quite a while. I think it's kind of important to ask these sorts of straightforward questions about what is pluripotency, and do you have different representations of pluripotency with different cellular acquisition routes.
Yeah.
And the paper that I think I want to draw everyone's attention to is in Stem Cell Reports from Derek van der Kooi, where he is making an argument that the neural crest is the sort of ground state. And I think that really comes back to the point that you and Flora were just discussing. If the people in this group are interested in this topic, I'd be very happy. Cheers.
And that was work in reprogramming, right? So when you're taking fibroblasts, what cells are actually getting reprogrammed? Am I remembering the van der Kooi correctly?
Yeah, it's really quite an ambitious set of statements he's making there, actually. I really recommend it to people. Yeah.
Cool. Thanks. So here's the panel of all 22 patterns where we looked at these two patterns in neural induction, and we looked at pattern 7, which was pluripotency, and then pattern 3, whereas pattern 3 was mesendodermal. So there was a lot of mesendodermal dynamics because, as I said, things move much more quickly when you put BNP on them than when you give them dual SMAD inhibition. So there's a lot of dynamics within the mesendodermal lineage emergence. And one other thing we saw, which really captivated us from the beginning, is 6 of these 22 patterns that the algorithm picked in order to explain what was going on in these dishes were invariant across time and almost invariant across condition, and simply a trait of the cell line producing that sample. So I don't know if you can tell, I know it's a little small here, but in each one of these, the plotting symbol is different across these plots. And there's 3 lines for each because there are 3 conditions and then the 3 conditions and 3 time points for each of those lines. So each one cell line in this collection of 22 pieces to put the whole picture together, each cell line basically has its own signature. Mm-hmm. And one of the things that made this, what made this interesting to us is this is a sort of a transcriptomic trait. It is stable across time. And one handy thing about the ES cell H9 is that many different people in the world have studied it. So let me check if I put so I didn't put the slide in, but I'll tell you it. We gathered RNA sequencing from 4 or 5 different labs done across probably 5 to 10 years of time, different sequencing technologies, and anybody on the planet who sequenced the H9 cell line, as well as other cell lines, when you take this pattern 13, the transcriptomic signature of the H9 cell line in our dishes, it is everywhere. Everyone who ever grows the H9 cell line, the H9 cell line is expressing this transcriptomic signature at higher levels than other cell lines. And what does that mean? If you remember back to what these patterns are, that means there's a whole packet of genes. The genes at the top of this pattern with the highest weights are simply expressed in the H9 cell line a little bit more highly than most other people. Because we don't have thousands of protein coding genes that one person can use and another person can't. We're all using the same pieces. And this is what we've been learning from complex genetics over years now, is that the difference between people, their risk for complex disorders, is in gene regulation. And this is a piece of what almost appears to be static gene regulation, that there are different set points or idling points for the expression of genes in the cells that all share one genome, that are from the same cell line. So now we wanted to bring these two things together. The biases we're seeing in these different cell lines' differentiation capacity, with the fact that we know they have these stable transcriptomic signatures that all of the cells in any condition are always kind of humming. Okay, let me get rid of these. So now we're going to focus on the two cell lines at the two ends of this spectrum, the SAO1 and I4 cell lines. What we're looking at here in black is the distribution of the weights in their gene signature. So there's a distribution of weights. And so if you have a high weight here, this pattern, or I4, is expressing it at high levels. So this GBX2 gene, which many of you may be familiar with, helps form the mid-hindbrain border in opposition to OTX2. It is very high in the I4 signature. OTX2 is not far behind, but in SAO1, it also expresses OTX2 at high levels, but GBX2 is quite low. So another assay that Sulky was doing on these cell lines, in addition to simply the neural versus mesendodermal differentiation, was retinoic acid posteriorization of the neural lineage as it emerges. So it turns out these two cell lines differ in that dimension as well. So in response to retinoic acid, the assay here is the number of HOXB1 positive cells. So these are very posteriorized brainstem down to motor neuron fate. And what we noticed is there's a rank order here where I4 is producing more of these posterior HOXB1 positive cells, and SAO1 and other lines are producing less. And if you actually plot their ability to produce HOXB1 cells across the enrichment of not just GBX2, but of a whole package of retinoic acid response genes, that's what's being shown here in red. I4 makes lots of HOXB1 cells and has lots of retinoic acid response gene expression. And the ability to make HOXB1 cells is directly related to how much at baseline you express these retinoic acid genes. It's important that the measurement of the retinoic acid response genes are not in response to retinoic acid. This is the retinoic acid response gene expression in pluripotency and the experiments we've done already, not retinoic acid experiments. So,
can I ask you, so there is no exposure to retinoic acid in here. There is just gene expression.
The
right?
retinoic acid assay we do to see how many HOXB1 positive cells they will produce. But and so that's the y-axis. When you give them retinoic acid, how much of a HOXB1 response do you get? The x-axis is not in retinoic acid exposure. That is in pluripotency and the two differentiation conditions that we just looked at. So it's actually the level of retinoic acid response genes in their cell line specific signature. But that was created without exposure to retinoic acid. So this is something these cells are doing in pluripotency and in early lineage emergence, even before they see retinoic acid exogenously. So it's like
this makes perfect sense, because OTX2 is actually more of an anterior gene that is repressed by retinoic acid that's published in mouse. And if you knock out OTX2, you get a headless mouse. Has the spinal cord, has everything, but beyond the MES-MET boundary, there is nothing. It's a very dramatic phenotype. So I would call this an anterior posterior bias, right?
Absolutely. Absolutely, 100%. And so what we're saying is when you put cell lines in a dish and expose them to retinoic acid, let's say we didn't know anything about these cell lines, we just knew that some of them responded less and some respond more. So, and that's, I think, people see lineage bias everywhere. It's been the problem, the challenge, and in our opinion, the thing we need to understand to make progress here is, well, how and why are they different? And as you mentioned, these two genes do a dance. If you look at the expression of these two genes in the brain, there's a line where they switch over that describes exactly the knockout phenotype that you just mentioned. But what we're saying here is that there's an idling process or a baseline level at which some cell lines express genes. For instance, IO4 has a lot of GBX2 expressed before it ever sees retinoic acid, before it's even being pushed regionally by anything exogenously. And the SAO1 line is almost expressing no GBX2 at all. So this is how I think of it as idling, or their steady state before going anywhere. You can already see where cell lines are tilted toward going, if you know what genes to look at to inform a particular decision. And as you said, the OTX2 GBX2 dance is well known in determining this anterior posterior game, so that when we look at this, we can understand it. But the wider thing to realize here is that in the broad pattern of the SAO1 or I4 or any pluripotent stem cells specific transcriptomic signature, again, this spans thousands of genes. So as much developmental biology and neuropsychiatric risk biology that we can understand, we can then start to leverage the fact that certain cell lines, and as we'll look forward now, in certain donors across multiple cell lines, express steady state differences in expression that in vitro we know bias these decisions, and in vivo we really don't know what they do. But Flora, your lab, as well as many others, have shown that some of these regional decisions are intimately intertwined with risk. So as we start understanding more of these pieces, I think it's going to become regular or standard, just as people are trying to sequence everyone's genome to get an idea of what we should do with that patient. We are going to be studying the cells of particular people. Now, that sounds a little bit like the current cancer treatments that are breakthrough and they're very tailored, but not scalable. But the understanding that comes from that, I think will really enable more of this. So.
Yeah, but we don't know whether this is the personal genome that does that, right? I mean,
that's right. But we can get a lot closer in the next couple. I'm going to skip over that for time here. I'm moving a little bit slower than I thought, but that's exactly what this next piece is. So now we used a new set of 6 lines. They're all IPS lines, and they have the extremely cool feature that we not only have 2 lines from this donor, 2 lines from this donor, and 2 lines from this donor, but we also had their fibroblasts and their postmortem brain tissue. So we put them through almost the same experiment as the other 6 lines, where we had self-renewal, neuroectodermal, and mesendodermal differentiation. And we did that NMF decomposition to break apart the pieces of what's happening. I won't go through the showing you that the same lineage emergence patterns come up, but what I will show you is the cell line patterns came up, and not only cell line patterns, but these are now individual patterns that came up that span both clones of a donor and only that donor across all conditions. So this is the 2063. Any sample from that cell line, no matter what the time or condition, is high in this pattern. Both lines, again, that being key. And we had that for each of the donors. So now it's only 2 per donor, but it's pretty convincing that these are now not about the cell line, but about the donor. And what really kind of put us over the top in believing that that statement was true is that, again, when we take a step to project things, this is now, each dot is a single postmortem human brain sample, one of which is, or 3 of which we also had in a dish. So if you look at the strength, what's on the y-axis down here, is the strength of the 2063 transcriptomic signature in the postmortem brain tissue. So that's here. This is the same 300 samples. The signature that we got from the donor 2053 in vitro, if you project it in vivo, here's how high its expression is, above all the other brain samples. So, and again, the third one is the same. Here it is way up here. So even in the context of just 3 humans, comparing 3 humans to each other with 2 lines each, the differences between those 6 lines, between those 3 humans in a dish, is very directly observable in the transcriptome of their 50 or 60, well, I guess it's 20-year-old brain tissue. So as far as
black dots, are they different regions?
The black dots, it's all one region. It's different donors. So this is 300 brain
different
donors. Yep.
300. Okay.
Yep. And so again here, the point was to show the expression of the transcriptomic signature that we derived in vitro in their in vivo tissues. And it's there, and I'm not going to show you the fibroblast, but it's same in the fibroblasts. The signature identifies the parental fibroblasts of the donor.
And Carlo, did you identify instead other pattern that instead are difference between clones from the same individuals?
So that's, yeah, I think you sound like I might have coached you on what question to ask. So that's the next figure. So,
no, it's just because it's perfect.
So
perfect.
In fact, that's why we colored them this way in the figure. Both lines from this donor behave similarly. Both lines from this donor behave similarly. And the 2 lines from this donor behave in a divergent pattern. And we'll see.
Okay, we are in agreement.
Yeah, so they're for sure. And I know your groups, the people that I'm speaking to right now, are acutely aware of both genetic and epigenetic routes.
I'm very acutely.
Yes, acutely aware of how line variation can pop up. So first, a little bit more information on how they're different. In 2 different assays, both the posteriorizing assay with HOXB1 and a dorsal ventral assay in telencephalic development, these 2 lines go in different directions. One of them is, and we'll see in the next one which is which. I can't remember offhand which is which, but when we see gene expression, we'll see it. One of them goes dorsal, one goes ventral, one is more posterior, one is more anterior. And each of these always go the same way as its other clone. So what happened between these 2 lines is an interesting question for us. So in that same experiment I just described, there was also a pattern that only identified one of the lines from this donor. So it had a pattern that identified both of its lines and its brain and its fibroblasts, something that is pinned presumably either to the germline genetics of that individual, or an epigenetic thing that was put in place at a very small number of cells in the origin of this human and stuck, but that's across all the cell lines and across the adult tissues. That's what we just saw. This is a transcriptomic signature that is very stable only in this cell line. If you look at its strength in the person's brain tissue, it's right in the middle. It is not expressed in the brain tissue. So presumably this line-specific pattern is something that the -6 line acquired since leaving the influence of all the other cells, whether early in development or in, this is from
a somatic CMV.
Yes, could be any of these, right? So we actually also sequenced the genomes of the different lines, and again, you guys are more acutely aware of how that can tell you things or not tell you things. We did not see any huge rearrangements that would be, oh my gosh, this, there's just a genome that got scrambled. It doesn't have any more mutations than any of the other lines when you're comparing them to the other lines from that same individual. And it doesn't have any specific mutations in genes we know play roles in these. That's not to say there wasn't a CNV that could have caused this, But that exhaustive search is something that's hard to do with a sort of an N of 1 kind of experiment. But what's important is it is a alternate epigenetic state, no matter how it came about. And here's where I actually already told you this result where the -2 line has lower retinoic acid enrichment in its sort of idling. The -6 signature specifically has higher retinoic acid enrichment in it. And again, that's in pluripotency before anything happens. And then when we do the retinoic acid assay to see how many HOXB1 positive cells they generate, the line that has epigenetically raised retinoic acid response gene produces more of those HOXB1 cells. And again, so we don't act, let's just keep going here. So now, Sung Mei then did some H3K9 trimethyl among other histone mark sequencing. And what the first panel is showing us is that if we take this 2053 specific signature and we project it into not expression data now, but this H3K9 trimethyl, which of course is the heterochromatin or repressive mark, specifically where the expression of genes is higher in the 2053-6 line, at those loci, the H3K9 trimethyl is higher in the -2 line. So at this point, it looks like the acquired change is in the -6 line, and it has lost H3K9 trimethyl at many loci, all of which or some of which should be captured in this signature. So this is a global thing when we look at the whole signature. And again, as I said earlier, that's hundreds, if not more, gene weights. But then we look super specifically at one gene we know that matters here, the GBX2 locus. And the H3K9 levels in the 2053 line are quite high, and they're almost gone in the -6 line. So we don't know mechanistically if we could relieve.
So can I ask you, sorry, this line is the same one that you showed in the previous slide with the other line that with regard to the responsiveness to retinoic acid? Can you go back? This is the 0523, right? 2053.
Yes. But so the retinoic acid response is right here.
Here.
Yeah.
Okay. So this was not highly responsive to retinoic acid, was actually the low responsive.
No, it's the high. It's this one. It's -6 is this guy, which is this
But
guy.
that's the line specific, right? So this is a subclone of that. The other clones of that person, of that donor, are different, right? The two.
Yeah. So this is the other clone from that human, that same person, and it has both lower. So there's two experiments here too. There's a high retinoic acid and a low retinoic acid dose.
Yeah.
In each case, the -6 line that has its own acquired signature is doing more retinoic acid response, both at rest and when we actually give it the retinoic acid. And that's the one that has lost the repressive mark at GBX2. Again, even before retinoic acid this is in self-renewal. Even before they see retinoic acid, GBX2 is ready to go. And in the other line, which presumably reflects more of the cells of that human because its transcriptomic signature was in the tissue of that human, it has GBX2 all wrapped up. So,
but I mean, this loss of signature is only in the genes that pertain to this pattern, right? Not for other patterns.
That's right. That's right. So it doesn't, this is specific. This difference in H3K9 here is specifically at loci that we got from the signature. So specifically at loci that the -6 line is overexpressing are the loci that has lost that H3K9 trimethyl. And you know, we don't know 100% that if we were to remove this mark at that locus or add it here, that this entire cell behavior would change. But given the trail we've gotten to come here, we're pretty sure that this is mechanistically involved. But again, this also means that there's other players involved. This is the aggregate signal across all the weights for this pattern. So there's hundreds of genes playing a role in this.
Yeah.
In this particular assay, in this particular decision, GBX2 is super likely to be central, but it's not
Yeah.
the only thing that's different between these lines.
You know, I just want to make a comment. You know, I think it's easy when one's really deep in the data like this for people to get lost. But a good reason to stay awake while this discussion is taking place is because what we're talking about here is exactly that switch that Flora was stressing earlier in this conversation. This is the forebrain hindbrain switch we're looking at.
Right.
So, and you can see it in all of these data when you go looking through this this H3K9 switch that's controlling this. It's not just a single gene. There's a whole, what do you want to call it? A neural regulatory network that we need to understand here. Anyway, pleasure to listen to you.
Yeah, the other elements that have changed in the, you know, it's not just a single locus that got flipped, right? It's a gear shift in that cell line. And how many other decisions it could be influencing. And this is something you and I, Ron, often talk about, is how complex is that cell space? Are there 5 gears? Are there 1,000 gears? Is there a gear for every locus in the genome? How do these?
I mean, how do these pattern coordinate their H3K9 methyl 3 signature to be altogether lost? Right? Because as you said, there are many genes in that difference.
That's right. Something swept across the genome and changed.
May I add something?
Not the whole genome, only the genes for this.
How would you call not the whole genome, but something that is widespread but specific to certain loci.
All right. May I add something about H3K9 trimethylation? I have 2 points that I want to make. Number one is that we are showing H3K9 in this one specific pattern. However, when we look at, in general, the identity patterns, the cell line specific patterns, what we see is that H3K9 trimethylation tends to be enriched in those genes. And number two is that it looks like in this pattern, almost all the H3K9 trimethylation is lost. But what you need to remember is that over 90% of the peaks of H3K9 trimethylation exist without in the outside of the genic region. So this is a small portion of the H3K9 trimethylation signature that exists in the entire genome.
Because this is in the gene, right? It's not
yes, we are looking at the TSS region specifically when we do these projections.
Okay.
Because our RNA-seq is gene-centric.
Yeah.
So we know the specific loci that 2053-6 is overexpressing, and then we looked at those loci for their H3K9. But as May is saying, that's a tiny proportion of the global H3K9 trimethyl in a genome, which is mostly non-genic or intergenic, right?
May, can I ask you, I think you always make these very interesting contributions on this topic. I'm being totally sincere. So what I find myself thinking is that other people must have seen this. And in fact, we've had a discussion where we talked about Joanna Wysocka work, where it looked like she was seeing something which was sort of eerily reminiscent of what we're talking about here. So I think this point might be something that we all want to sort of put on our agenda to really come back to. These are not, when we come across them, they seem very special. But as you're pointing out, they have these very special qualities, but they're very biologically significant. Other people are seeing things like this.
Yeah, thinking about your comment, May, that most of the H3K9 is not where we're looking here currently. I wonder if there's a way that if we had some hook to look more globally at H3K9 at what was the global switch that happened, what and where got flipped and what and where did not get flipped between.
There are papers now. Well, this is histone methylation, but there are papers at single cell DNA methylation level that distinguish across different cells and different lineages and different cell types. So it will come up. I mean, I don't know. I mean, how efficient would be to do single cell
Well, I think that the DNA methylation would be a great place to start
Yeah,
looking.
right.
Again,
It
because
would be a good way to address this,
yeah,
yes.
yeah. Okay.
And one more question. In this
Yeah.
pattern 17, there are like retinoic acid genes enriched in this pattern, and they also are influenced by, are depleted of the K9 3 methylation.
Sorry, I missed your question. Can you say
Oh,
that again?
sorry. I just say in this pattern 17 that distinguish this line are included some retinoic acid responsive genes, elements, that they also follow GBX2.
Absolutely. So in addition to this, where we see at the GBX2 locus that the H3K9 trimethyl is gone, it overexpresses GBX2. So GBX2 is one of the highest genes in this pattern, as well as a whole packet of those retinoic acid response genes. They're all up. So it seems to be a concerted shift of a specific subset of the genome that's being switched. And again, we don't know exactly how this happened, whether it's a genetic aberration or a change in the epigenetic state, but it was a big gear shift.
Interesting.
Okay.
Hi, Carlo. It's Alex.
Hey, Alex.
How are you?
Good.
It's I wanted to ask you if I forgot if you tested also the effect of passage numbers and things like this. Like if you look at like more if there was some either some patterns or some explanation of why these lines.
So yeah, so I'm trying to think back to actually the response to reviewers we had. Silky had a whole portfolio of experiments. I don't know if Silky, feel free to jump in. She did dosing of dual SMAD. She did, I don't think passaging was one of them, or it might be. Do you know offhand? Is Silky or May, do you remember?
Actually, we actually didn't include the data, but we actually did some experiment with the multiple passages. The key result was we checked the SOX21 expression level across the cell line with different passage number, and we've been seeing same bias. That's the
Great. Yeah,
So it's
so
quite robust against culture. So it's good.
that's right. Yeah. So time and dosing and I did not remember, but as she just said, is passaging. The cellular phenotypes that were different across those lines were incredibly stable. And of course, that's not to say that all cellular phenotypes are. Of course, that's one of the big problems now is that they aren't. But understanding which of these elements are stable traits of cell lines and of cells from particular donors is a huge thing that we need to be doing right now to understand this variation.
Yeah, no, it's really cool.
Great. Okay. So I realize we're a little late here, so I'm just going to wrap up the first story and not touch the second one. I'm very happy to talk more with you guys individually or as a group again, but this is from the last figure in the paper, where we've in our own new data generation had two sets of 6 lines to look at these differences and changes sort of at a high resolution of what individual cell lines are doing. Here we took a public data set from Ihor Lumishka's lab, 101 donors with multiple lines per donor, so that I think the total number of lines was right around 300. And they had bulk RNA sequencing and pluripotency for all 300 lines across the 100 donors. This is a plot of donors on the x-axis ranked by their PC1 in this data. So if you do PCA analysis of the 300 donors, this is just those donors ranked along the PC1. Now, what we're showing here is the level of expression of retinoic acid response genes in each of those cell lines. And as you can see, the PC1 correlates greatly with retinoic acid gene response expression. It corresponds with the PC1 of our cell lines. The difference between I4 and SAO1 is along the same axis. What was something I've highlighted here also, just as we saw one line behaving very differently than another line from that donor, any dots on the same vertical line here are from the same donor. That's why they're donor ranked. So this donor had two lines that were down here and one that was up here, higher in PC1 and higher in the retinoic acid. But in general, what you notice is that there's very little vertical variance outside of a couple of these. There's a couple up here, maybe a couple down here. But in general, if your first cell line is down here, your second cell line is down here. If your first cell line is here, your second cell line is up here. So the genetic effect is much stronger in general than the epigenetic effects, or at least the...
Well, thank you for showing this slide, because this is the, yeah, I didn't remember. Did you have this in the paper or?
Yes,
or?
this is in the final figure of the paper where we basically
Okay, that's
show
very nice. So
that,
it does show that in fact the donor effect is stronger, right, across 300 lines.
That's right.
Which is
And
pretty
this is sort of a formal statistical calculation of that. This is the interclass correlation metric, which is really just looking at the within donor variation versus the across donor variation.
Yeah.
And it's the PC1 and retinoic acid gene expression is highly donor dependent. And so we, in a number of analyses, we show that the primary dimension of variation across these 300 lines is exactly what we bumped into in our small sets of 6 lines, where we got
so anterior, posterior, right? And it's agree with us with our chamber results, Soraya's and Alex's paper, that when we tried the 2 orthogonal dimension, sonic hedgehog and ventral, dorsoventral, and wind anterior posterior, the dorsoventral was not as variable across line, across subject as the anterior posterior. Yeah.
Super interesting. And it would be interesting if I think you have RNA-seq from pluripotency in those lines as well, correct?
Most of them. I'm not sure we have it from all of them.
That would be great.
Look at that.
We could very easily see if the prediction still works, where you have your
making this plot here.
Yeah. And then we have some metric of retinoic acid gene response in pluripotency on this line, and I'm guessing we would it would be wonderful to look at your data along those axes.
Right.
But again, the key is that the predictive axis isn't what it did molecularly when it saw retinoic acid. It's how much of the retinoic acid response was already cooking before those cell lines saw retinoic acid. And so that's the left half of this. The primary direction of variation in the 300 lines is very parallel to that which we saw in our small number of lines. And if we take this dimension, this PC1, the difference between pluripotent stem cell lines in a dish, and we project that onto the map of mouse development, where purple and pink is low and yellow is high, we see that the pluripotent epiblast is super low, so toward that end. And in fact, that low is actually preserved for some time as cells move toward the neural lineage. But if you move toward the mesendodermal lineage, well, the mes or endodermal, which is down here, it turns up quite quickly. You go through the purple, black, all the way to yellow here and here. So basically the bias in pluripotent stem cells in self-renewal, this is all self-renewal, there's no differentiation in this paper. How pluripotent stem cells balance themselves, that variation is along the axis of lineage emergence in the early embryo.
That's super interesting. And that's largely donor specific. That means that donors are slightly different in the way they pattern themselves at the early embryonic stage.
That's right. And it's and I think my next slide is just let's think about this slide. Let's see. Yeah. So I think this is a perfect point or in the talk to go through this. And as I put up these points, please interrupt and discuss as you wish. But putting together all the things we've just been talking about, we and many people have seen these biases in lineage commitment. What we do here is really sort of map it precisely onto development, but also note the existence of transcriptomic patterns that are specific to both lines and or donors, which I think, you know, if you look at the data in the world, it's out there, but it's not what people have been looking for. And in fact, we've spoken to some people who explicitly try to avoid this because they're looking for low variance assays. But then these stable transcriptomic patterns determine the lineage bias. And then on top of that, it looks like some of these patterns, if we can break it apart correctly with the number of clones in mature tissue, we can tell a line-specific pattern from a donor-specific pattern. And these donor-specific patterns, as far as we can tell, are expressed in every cell of that human throughout its life. There's no reason to think that its skin cells and brain cells and pluripotent cells are doing something other than its heart cells or that person's heart cells. And we have some GTEx data actually to suggest that. The problem with the GTEx data, though, is each human comes from a place, so there's some confounding there. But in any case, it really looks like there's not a question. There are stable transcriptomic patterns that are traits of individual human cells. Now, if we say, well, that biases their pluripotent stem cells and how they differentiate in a dish, how then can we use that? What does that mean about the human cell biology? If I have a tumor and my personal transcriptomic signature is different than somebody else, can we use that signature to predict either the prognosis for that tumor or how best to treat that tumor? As an example, something that I'm sort of trying to revive is an observation we made in this study where two of the blue patterns, the BMP patterns, we looked at that one pattern 3 that was different across cell lines. They're very different across cell lines. And two of them, if you look at their levels in GBM tumors, in glioblastoma tumors, they predict the severity and aggressivity of the tumors. So that means that BMP driven or TGF beta signaling driven transcriptomic networks that are used in the first steps of lineage emergence, if you look at those in a GBM tumor in a 50, 60, 70-year-old person's head, how hot one or the other of those patterns is in that tumor predicts their survival by years. So it's really a case of, I think, leveraging the new, which is all these things we're talking about seeing, which is the old, and the canonical signaling pathways that we know control development, that we know control cancer, and the realization that with not too much of a crazy amount of studying someone's cells in a dish, as long as we have other people's cells to compare them to, we can start to glean seriously powerful information about how that person's cells may react in X, Y, or Z context. And hopefully, in the not too distant future, use that information to educate how we diagnose and treat patients. And I know, I think it's not, when I say it, it sounds like a big jump, but logically speaking, conceptually, I think we're there. The question is, how would we study many different human cells deeply enough to understand enough about their biology and their stable transcriptomic signatures, their stable cellular phenotypes to do this? And how could we do that in a cost-efficient way, which is a hugely wide open question right now. But people have made the repositories. There's plenty of people collecting lines. I think now together we have to think about what to do with them to really get value out of studying hundreds of people's cells. So I'll stop there. I think we're over time. I thanks so much. And if you want to know more about the NEMO system, I'm very happy to talk to you. I've talked to Flora and Alex and Soraya quite a lot about it. But putting all the data we just looked at, surfing those different data types and saying, okay, look at this signature in that data set, that model is really what we try to leverage with the NEMO system.
Yeah,
Thanks
I
so
mean,
much.
we're thinking to write a grant on this to specifically look at variation. Sounds technical, but it's not. And across all our lines, I mean, we have more than 1,000 now.
Ooh.
And that would allow you to look at this question I was asking you at the beginning, whether variation is higher in patients than controls. I don't know. We would have to see, even if it's 1,000, mean, 1,000 including clonal variant for each
Got it.
person. So, yeah.
Which is great that you have a lot of that's not always included in the structure. And I think even if it's a small number of replicates, 1 or 2 per person,
Yeah.
as long as you have them in many people, that will enable you to do the bulk of what you want. You don't need 20 of one person. You need many replicates of many.
I love that plot that you did for the Lemska lines with 300 lines. That was only at the IPS stage, but you know, in principle, one can do also for earlier stages. Yeah, thank you. That was very interesting. Are you able to share the slides? If you can,
Absolutely.
we can send them by email to all the
That's
participants.
perfect. Yeah. So, and what I'll do right in the slides, I have these are actually live links. So if you play the PowerPoint presentation when I send you the slides, you can click on this and it will take you to the live NEMO page with this data. And it's something I haven't mentioned, but I know a lot of you are aware of. This work together, Soki, Sungmei, and Ron and I really kind of inspired us to start the Developmental Diversity in a Dish seminar series that you've played a wonderful role in, Flora. And so this is a link to the site where you can look at what seminars are coming up. And in fact, it's next Tuesday is our next one.
Okay.
Thanks
Thank
so
you
much.
so much. And I know that there was Yon John that wanted to present at the last moment. I'm sorry, I didn't know that earlier, but do you think you could do it next month, the next meeting?
Sure, I'll do that.
Okay, great. Okay, thanks everybody. Thanks, Carlo, of course.
Thank you.
And I'll see you at, Carlo, by the way, if you want to participate to these meetings, you're welcome to. We have them once a month. And
yeah, stick me on the
I'll send you the agenda in case you're
Great.
interested.
Really appreciate it. Would love to. Thanks.
Okay. Bye. Bye,
Grazie. Ciao.
everyone.
Thank you. Bye.